This second video about viruses is all about host range, and I will conclude with a tiny bit about viral size. So host range refers to the spectrum of host cells that a virus can infect. We typically describe host range as narrow if a virus can only infect a limited number of hosts. So for instance, most bacteriophage, which are viruses that only infect bacteria, most bacteriophage can only infect one species of bacteria. So it has a very narrow host range. Some animal viruses have a very large host range. We typically use the term broad instead of large. So a broad host range means the virus can infect a lot of different organisms. For instance, West Nile virus can infect horses, alligators, sheep, all sorts of things, like even alpacas, right? So they can infect a lot of different organisms, so they have a broad host range. So what determines the host range of a virus? Well, typically it's going to be the receptors that the host cell has on its surface that allows molecules, to come inside. And so the virus essentially has proteins on its surface, which we will call spikes, that mimic the shape of that host cell receptor. And so they essentially trick the host cell into thinking that they are something the cell needs. And so the viruses are able to attach to these host cells and then, as you'll learn in future videos, gain entry into the cell one of a variety of ways. So the virus has a protein that is specific to a receptor found on the host cell. Okay, so let's go ahead and sketch this out. There's going to be quite a few drawings that we do in these videos because sometimes just looking at a complex figure can actually make things harder to understand. So I want to really break it down either by animations or drawing some of the things myself. And I'm still using my mouse pen, so I apologize that this won't be quite as pretty as it perhaps could be. So let's go ahead and draw 3 different possible host cells. So this is going to be us, so human. I'm just going to abbreviate HUM, a human. Now let's go ahead and draw us a hippo cell, right? My favorite animal. So let's include them here. And then let's do my second favorite animal, which is going to be a sloth. Okay, so on the surface of cells are various receptor molecules, and they have all sorts of different shapes and sizes. And again, they're specific for molecules that the host does need. Okay, so the virus is just being tricky. So let's pretend that on the surface of human cells, I'm only going to draw 3, but please note these would be completely covering the cell. Let's go ahead and make this one kind of like a box. Square in shape. Let's say on the hippo, it's more like angled, like a triangle would fit here. And then for the sloth, let's try and do more like circular. Sometimes I have a hard time doing that with this mouse pen, but these are supposed to be more like round in shape. So just please make sure that they look very different than your human. You can pick whatever shape you want, really, but we want each of these organisms to have very different shaped receptors for this particular example. So now let's say we have this virus, okay, and it's going to look like this. It's called a icosahedral shape, and we'll talk more about that in a different video. But a lot of times coming off of this virus are going to be these proteins. We call them spikes for obvious reasons. They look like spikes on the virus, and they have a specific shape. So I'm only going to draw 2 of them, but please on the end of each of your spikes, draw this triangular shaped region. Okay, and again, it's on all of them. They should be pretty small. I'm just having a hard time with this pen. Okay, but there'd be that triangle shape on the edge of each of these lines sticking out of the virus. And so what would happen is this virus is going to come into contact, with different organisms perhaps, but is it actually going to be able to infect them? Well, that depends on the specificity of the virus, and that's the specificity means the specific shape of the viral protein must be complementary to or match the host cell receptor. So can this triangle viral protein easily and firmly attach to these round sloth receptor sites? No. Can this virus, can its triangular viral proteins easily and firmly attach to these human square receptors? No, right? They're not complementary. They'll just kind of like fall out, right? So then, can this viral triangular shaped protein attach here to the triangular shaped receptors? Yes. Okay, yes, they can, because they are complementary. So I'm going to actually erase these lines now so that I have more room. And so what's essentially going to happen is this virus here is going to attach. Its viral protein is able to fit nicely into this hippo receptor, and it tricks the hippo cell into letting it in. And we'll talk about mechanisms of entry in a different video. Okay, so this is what we refer to as the specificity between the viral protein and the host cell receptor. Okay, so we are actually nowadays trying to target different diseases and things like cancer using phage therapy, or we're considering trying it depending on the disease and things that we're talking about. But the idea is pretty nice and promising, right? So phage, we refer to bacteriophage as phage, okay, not animal viruses. And again, bacteriophage are viruses that can only infect bacteria. And again, because their receptors are very specific, okay, the viral proteins have a very unique shape that only can attach to those very specific receptors. So what we can do is look at infected cells and find a receptor that is unique to only the infected cells. And then we can manipulate a bacteriophage so that it can only recognize and attach to the receptor on the infected cells. Okay, so there's that specificity that we need. Only infected cells would the phage be able to attach to. So that means it wouldn't harm the other healthy cells that the host needs. And then instead of delivering viral nucleic acid, right, because nobody wants that, it would be delivering some sort of therapy or drugs to that infected cell, in theory, to kill it and eliminate it so that the person won't be sick anymore. So just little tastes of some things we're going to talk about later. We can talk about how microbes are used to, in various areas of life. So phage therapy is kind of going on this idea of host range to make sure that we're delivering drugs, for instance, to a specific infected cell. When it comes to viral size, right, viruses are tiny, tiny, tiny. I had a college professor that, as to kind of give a visual, said, imagine the sharp pointy end of a safety pin, right? It's really small. Now, some viruses, now they vary in size. Okay, they do range from 20 to 1,000 nanometers in length. Nanometers, that's tiny, y'all. So she said, if you think about the sharp end of a safety pin, some viruses, they could fit 1 million of those viruses on the tip of that safety pin. That's how small they are. Okay, so they are super, super small. And so where does this apply to y'all's daily lives right now? Well, masks, right, with coronavirus. Coronavirus is pretty small. It's about 100 nanometers or 0.1 micrometers. A lot of times we talk in micrometers. So COVID is about 0.1. We also say microns instead. So I'm going to use that term. It's just easier to say. So COVID is about 0.1 microns. And N95 masks protect they have pore sizes are small enough to protect anything in size about 0.1 to about 0.3 microns. So COVID does fit within that range where it is going to be able to protect and prevent a lot of virus from getting through that mask. Okay, so they do on N95s offer protection with about 95% efficiency. That's why you see some people wearing those, because you're really keeping yourself and other people safe because the virus just can't get through that mask. Surgical masks, they range in size, I believe, from about 0.3 microns to 10 microns. So their pore sizes are much larger. Okay, COVID can fit through them, but do remember that the virus is coming with respiratory droplets, right? When you cough or sneeze and things. So they are a bit bigger. But masks are still a really good way to prevent some of those big droplets and other things from getting in and out of the mask. Masks also have it's like 3 different layers to them. Like the inner protection, or the inner layer, like facing your mouth, is very absorbent, right? So it's absorbing those respiratory droplets when you cough, sneeze, talk, laugh, giggle, whatever. That could be carrying COVID or other things. And then the middle layer is actually where the pores, the actual like filter is at. Then there's an outer layer that's meant to repel liquid, right? So if somebody's talking at you, it's meant to not soak through and just offer this extra layer of protection. So wear a mask, y'all. It is scientifically proven that they are efficient. I mean, it all comes down to pore size and virus size. So we're going to talk more about this concept in a future video. All right, y'all, let me know if you have any questions.